Thank you for the introduction. Hello everyone, my name is Wanxin Li. I'm a PhD student from the University of British Columbia in Canada. So today I'm going to introduce our work on using a Riemannian elastic metric for statistical analysis of tumor cell shape heterogeneity. This is a joint work with Ashok, Nina, and Kong. So in this presentation, I'll start by introducing the motivation of the problem, and then I'll describe our method, and then I will go over the dataset and results and draw conclusions in the end. So let's first start with the motivation. The culture cells on planar surfaces usually has a variety of shapes, and the shapes are usually affected by the molecular processes happening on the cellular membrane or cytoskeleton. The relationship between the cell shape and the molecular processes suggests various potential applications of quantitative measurements of cellular morphology. And among those measurements, biophysical measurements using cell elasticity on tumor cell shapes have shown some heterogeneity across cell types, cultures, and measurement conditions. So the dataset we use was previously used to develop an image processing and data analysis toolbox called tsmorph. It is a tool to quantify the texture, irregularities, and the spreading of single cells. And the dataset contains two murine osteosarcoma cell lines, DOM and DLM8. They differ based on their metastatic potentials. And the control group was treated by dimethyl sulfoxide. The treatment groups were treated by just quinolide and cytochalasin D. So cytochalasin D binds to actin and prevents the actin film and prevents the actin polymerization. So it thus leads to actin depolymerization. On the other hand, just quinolide also is an actin-binding drug. However, it stabilizes actin polymerization, so they have reverse effect on the cellular morphology. So here's a brief overview of the elastic metric for cell shape analysis. In 2007, Mill proposed the elastic metrics to compare curves. So the elastic metric is defined to be a Riemannian metric defined on the space of curves, and a Riemannian metric equips the manifold with a smoothly varying product on every point of the mani-curve. The elastic metric quantifies the extent of bending and stretching to morph a curve into another one. And it has been applied to a variety of biological shapes, including biometrics and human body shapes. Recent developments on elastic metric include efficient computation of the geodesics of the elastic metric. So here are our research goals. Firstly, we want to use elastic metric to quantify the local deformations of a curve by evaluating how they bend and stretch. And secondly, we want to apply elastic metric to tumor cell images to investigate the elasticity of cell shape and conditions. And thirdly, we want to demonstrate the better performance of elastic metric over its competitor, the linear metric, on tumor cell images. Okay, so let's now introduce elastic metric mathematically. So suppose C is a smooth parameterized curve. H and K are two curve deformations in the tangent space of C, so which can also be considered as planar curves. And the N and T here are two local unit normal and tangent vector for the moving frame associated with C. And with the A and B greater than 0 being the parameters of the family, one can associate every curve C with an inner product, which is defined to be the elastic metric by the sum of two components and given in the following form. So the first component here is the projection of the derivative of h and k onto the local normal vector of C. And it can be interpreted as how much to bend the curve. And the second component here it is the projection of the derivative of h and k onto the local tangent vector of C. And this can be interpreted as how much to stretch the curve. So intuitively, elements in the tangent space in C represent infinitesimal deformations of C, so namely here h and k. And the elastic metric quantifies the magnitude of these deformations with two factors A and B that can be interpreted as the weights penalizing the cost of bending and stretching. And when A is 1 and B is 0.5, the elastic metric becomes the so-called square root velocity metric, which is efficient for evaluation. And next, we define alpha to be a regular parameterized path starting with C0 and ending with C1. So we can interpret alpha as a prescription for bending and stretching the curve from C0 to C1. And we define the length of alpha associated with the elastic metric to be in the following form. So it's integrating the elastic metric over the velocity of bending and stretching on the unit time interval from 0 to 1. And then the geodesic distance between C0 and C1 is defined to be the minimum path between C0 and C1. And the figure on the right-hand side shows an example of a path between C0 and C1 by the gray curves in between. And when the path is the minimal path, it is defined to be the geodesic between the two curves. It has been recently shown that upon a transformation that maps the geodesics into straight lines, the geodesic distance can be computed as the L2 metric of the two transformed curves. While the procedure to find such mapping for a general elastic metric is numerically unstable, it is simple for the SRV metric. So we can write the geodesic distance for the SRV metric in the following form as a pullback of the L2 metric onto a transformation of the two curves. Okay, and then the Frechet mean of n curves is defined to be the centroid of n curves. We also provide the definition for the linear distance for two curves and the linear mean of n curves, as we chose the linear metric as the competitor for the SRV metric. On the right-hand side, we show a comparison between the Frechet mean and the linear mean. So we can see that the Frechet mean is an element belonging to the manifold, whereas the linear mean is not an element belonging to the manifold. Okay, so let's now talk about the dataset and the results. We use the implementation from GeomeStats. So GeomeStats is an open source Python package built for machine learning for nonlinear data, a field called geometric learning. So it provides object-oriented and extensively unit-tested implementations for essential manifolds, operations, and learning methods with the support for different execution backends, namely NumPy, PyTorch, and TensorFlow. And as we mentioned before, our dataset contains two murine osteosarcoma cell lines, DOM and DLM8. And here we only present the result for the DOM cell lines. We pre-process the data by removing the outliers using dcore MDS. So dcore MDS is our recently published tool based on the geometry and the statistics of the simplices formed by data points, which allows to detect orthogonal outliers and reduce dimensionality. So after the removal of outliers, the DOM cell lines has around 400 cells with 200 cells in the control group and 100 cells in each of the treatment group. And as we discussed before, jasperkinolide and cytochalasin D are two cancer drugs that differently affect the cell morphology. as cytochalasin D disassembles the actin filaments and causes the cytosolic aggregation of the actin masses. The resulting loss in the internal tension forces is expected to give rise to shapes with many invaginations, with increasing distance from the smooth ellipsoid. On the other hand, the effect of jasperthichinolide is less dramatic as it leads to the formation of a more diffuse actin network, but without affecting the stress fibers. So we first studied the result by visualizing the distances to the mean shape for different conditions using the linear metric and the SRV metric. So as highlighted by the red curves, we observe that the SRV metric detects more irregular cells for the 90th percentile, while the linear metric detects less regular cells for the 0th percentile. So this indicates that the SRV metric captures more irregularities as percentiles go up. And then we study the result by visualizing the histograms of the distances to the global mean using the linear metric and the SRV metric. So there are several observations here. So firstly, we observe that using the SRV metric, the distributions under different conditions are more well separated. So here you can see that the distribution for the blue curve and the yellow curve and the red curve and the red distribution, they are more separated than they are in the other one. And the second observation is that the distribution of cytochalasin D, so the distribution represented by the red one, is wider than that for the distribution of jasperthichinolide, so the one represented by the yellow distribution. And this aligns with the biological mechanism that the effect of cytochalasin D is more dramatic than jasperthichinolide. And the third observation is that using the SRV metric, we notice a bimodal distribution for the control group here. And this potentially suggests multiple cell states within the existence of multiple cell states within the same cell line. So this analysis shows that the SRV metric yields a better separation across different conditions. And we finally study the result by projecting the data onto lower dimensions using multidimensional scaling, where the distance is defined by the linear metric or the SRV metric. And the results suggest that the SRV is better at capturing the cell heterogeneity, as we can observe that most of the control cells are located in the middle, surrounded by jasperthichinolide treated cells, and cytochalasin D treated cells being located even further. We also examined the stress statistics obtained for the multidimensional scaling for all the dimensions tested. So in figure C, we observe that the SRV always achieves a better stress statistic than the linear metric for all the dimensions tested. And we note that in order to produce figure A and figure B, we chose dimension 2. And we can see that in figure C, in dimension 2, the SRV stress statistic is significantly than that of the linear metric. So this analysis shows that the SRV metric captures more informative patterns when it comes to dimensionality reduction. So in this study, we used the SRV metric to statistically analyze the 2D shapes from cell images. And SRV consistently showed better performance over the linear metric for all the tasks we analyzed. And specifically, it shows better performance at capturing the cell shape heterogeneity. It provides better separation between different cell groups, and it provides better low-dimensional representation when it comes to dimensionality reduction. And in the future, we want to benchmark the SRV metric against the metrics including derivative information, such as the Hessian metric. And secondly, the development of the elastic metric as a tool for practitioners would require more thorough investigation of morphological changes observed for different cell types and conditions, and jointly with other features existing in the literature. And thirdly, we want to apply this SRV analysis to a larger dataset consisting of cheek embryocardial fibroblast time-lapse images. And this dataset was collected by our collaborator at Shork's lab. A video of the dataset was shown below. So you can see that it's a dataset of different timestamps, and at each timestamp there are multiple cells. And finally, we want to generalize our dataset to the whole family of elastic metrics. And this brings me to the end of my presentation. I want to thank my collaborators for their insightful inputs to this project. And if you have any questions, comments, feel free to ask. Thank you very much.
Thank you for this nice and deep talk. So have you got questions? Are there some questions?
Thank you for the presentation. Could you give maybe some details about future works, in particular the comparison with metrics including derivative information? Do you have ideas about this point?
Oh, sure. So one of the limitations of our work is that right now we only compare with the linear metric, which is a relatively simple metric, and we want to see that whether the better performance of the elastic metric comes from it contains the derivative information. So that's why we want to compare it with a metric that contains derivative information, such as the Hessian metric. Yeah.
Okay.
Does that answer your question?
Yeah.
Okay.
Some other questions?
Thank you for the good presentation. I had a question regarding the slide where you show the histograms of your different cell populations and the distance to the mean. That seems, if I understand, it's just like the spreading around the mean is different. Is that the idea? Like the controlled cells are more coherently are closer to the mean in general, whereas the CITD cells can be anywhere but further away. Is that what they say?
Do you mean this one or this one?
No, I was talking about this one.
Oh,
maybe I
okay.
didn't understand, but since you say it's distances to the global mean, what I read is that like the red population is further away from the global mean, but I find like I imagine a sort of ring or sphere that would be further away from the mean. Is that what happens? Like, are they in? Is it just the distance that's bigger?
It's just the distance that's bigger. So we compute the. So first we compute the. So we compute the fraction mean of all the curves, and that becomes the global mean, and then we compared every individual cell to that global mean using elastic metric, and it's because the ones in the treatment groups, they are affected by the two drugs, and we know that the cytochalasin D has a more dramatic effect on the cellular morphology. So ideally, it will have like a larger distance from the mean.
So distance from the mean means like what does it mean concretely? Like that the shape of the cell is more star-like or something like that? That it has more folds? Okay.
Yeah, it's like more irregularities from the
so
mean.
It's okay.
Yeah. Yeah. So for this slice, it's the mean for is the mean for different conditions individually, and for this one, is the global mean.